6, 5, 4, 3, 2, 1.
Hey, what's up, everybody? Welcome to this episode of NASA in Silicon Valley Live for September 13th, 2018. I'm your host, Abby Tabor, and I have with me a new face today, my co-host.
Hi, I'm Christina Chung, and if you didn't know, this is a NASA Silicon Valley Live, a conversational show out of NASA Ames Research Center with the various scientists, researchers, engineers, and all-around very cool people at NASA where we talk about the nerdy news that you need to know about.
Absolutely, yeah. And you can watch us live on Twitch, YouTube, and Facebook. But if you want to ask questions to our experts, who I'm going to introduce in a minute, you need to be on Twitch. So go to twitch.tv/NASA. Watch us live, but if you miss us, you can watch us on demand later, including on NASA TV. And there's always the audio version if you want to hear the podcast version. So let's go. Today, we're going to be talking to you about studying life in space, as in what happens to Earth life when we bring it into the weird conditions of space. And later on in the show, we're going to share some genius space hacks that our researchers have come up with, really simple solutions to do this incredible research that they're getting done. So we're going to get to that later, but first let me introduce you to our guests today. I have with me Sid Sun. Welcome. Sid is the manager of the Space Biosciences Division here at NASA Ames. And I have Sharmila Bhattacharya. Hey. She is a senior scientist here studying life in space. Welcome, guys. Now, I'm going to be looking at the chat for questions from the audience, so while I'm browsing there, meanwhile, Christina, do you want to get us into the science today?
Sure, definitely. Alrighty, so welcome, Sid and Sharmila. Thank you for coming here. And just to get all of us situated, what is space biosciences, and why is this important that NASA does space biosciences research?
Yeah, so we are studying life in space. We're looking at all different types of organisms, ranging from tiny little microbes, tissues, cells, fruit flies, other types of animals. And the reason we're looking at all these is that we want to understand the risks that are facing our astronauts as we go on to Mars. Mars itself is going to be roughly a 2 or 3 year round trip mission for our astronauts. There's going to be a lot of risks that they're going to be facing. There's a high radiation environment that they're going to be exposed to. They're going to be experiencing zero gravity as they're getting to Mars. When they get onto Mars, they'll be dealing with roughly a 1/3 gravity environment relative to what we experience here on Earth.
Oh, wow.
And then, of course, they have the long trip back. So there's a lot of changes they're going to be experiencing, and there's a lot of things that it's going to be hard to study on the astronauts themselves. So we do our studies with mice, fruit flies, and other organisms.
Oh, very cool. Sharmila, do you want to maybe expand on that a little bit?
Yeah, so I was going to say that, some of this, as we just talked about earlier, that you want to know what happens in the humans or in astronauts, but it's very hard to do these experiments on humans. And so you need these little surrogates, so to speak, these tiny organisms, so you actually understand the basic biological changes that are happening inside the cell, so that you can then send, with your crew that's going out there into Mars or Moon explorations, you can send them with, the medications they need, some of the countermeasures, and to know what to send and how to keep them healthy out there, you need to understand the basic biology underlying these changes, and that's what space biosciences is about.
Yeah, amazing. I already have a question. You can tell me if you think we're going to answer this throughout the show, but Rev Lucky Shot wants to know, what's the biggest difficulty in performing these tests in space?
Very good question. So for me, I think the biggest issue is the fact that you're limited in mass and volume. So the size of your experiment has to be, and you can't expect very much help from the crew because they're busy doing other more important things sometimes. And so you have to have it often be automated, it has to be small, and yet you're going to have to ask those important science questions and you want to make sure that when the experiment comes back, you can answer them. So designing that experiment so that it's successful, useful, but also fits within the constraints of space flight are the challenge, I think.
Oh, well, speaking of things that we need to send into space, I think we're going to move on to a Let's Play.
Our first segment.
Yes, our first segment, Let's Play, and we will be answering the question, has it been to space?
Yeah. You want to roll that
Bring it
segment,
on.
Bill? Get us ready.
There we go.
All right, guys. This is a game. I have some questions for you, and you guys watching can play along at home. And if you get, let's say, 10 out of 10 of my questions, we'll give you a shout out. So, that's a prize.
Right, no, I mean, shout out from NASA. I think that's pretty cool.
Yeah. So the question is, has it been to space? And I have for you 10 organisms. All right. The bear.
No, definitely not.
No, definitely not.
Surely not.
All right.
No polar bears, no
polar
brown
bear?
bears, no black bears.
All right. What about the jellyfish?
Jellyfish. Yes, we have flown jellyfish.
For real?
Yeah.
Why would you fly jellyfish?
Exactly why.
Yeah, well, it turns out jellyfish have these tiny little organs that help them sense the gravity field that they're within. And so we wanted to understand what happens when they're in an environment without gravity.
Fascinating.
Yeah.
That's cool to me that an ocean creature has that kind of.
Was it
sensing of
dangerous to
gravity.
set? I mean, I'm scared of a jellyfish sting.
Yeah.
I'm just saying, you know,
Gotta
that
stay
just
away
seems
from
like
those stingers.
right. That's- oof.
But that's where it's nice that it's all contained.
That is true.
True.
Very true.
In their tanks. All right. Ready. Rodents.
Yeah.
Definitely.
Yeah. In fact, that's
Most definitely.
one of the organisms we fly the most to do our experiments with.
Here at Ames, even, right? All right. Here's an odd one. Sorry, can you see it on the screen, or I can show you guys?
That is an odd one.
It's a squid.
It's a squid.
Yes.
Yes, we have done experiments
Really?
with squids.
So another ocean critter we've actually sent into
Yeah.
space.
Yes.
Squid, though.
No, not giant squids. At least.
Oh, good. I was going to say what
We
type of
flew
squid.
Tiny little squids.
Do you know why?
Well,
can you
it was
study?
an interesting experiment because these tiny squids, as they were growing from their baby stage, essentially, they were going through some changes in their body. And we wanted to understand those changes would be different in the environment that they're experiencing in space.
Oh.
Yeah.
Growing and developing and
Yeah.
how space changes that. Yeah. Cool. What about
No.
No eagle.
the eagle? No.
Our nation's favorite bird.
That's right.
He didn't get a chance to go? Oh, that seems unfair.
Too big.
All right. Those flying creatures know. What about
Yes.
the fruit fly?
The fruit fly, definitely.
Yes.
Sharmila and I both are kind of cheering right now. It's
Yes.
definitely our favorite.
Your favorite organisms?
Yes, indeed.
Okay, here's one of my favorites.
Spider,
Yes.
Yes, that's also
Arachnids.
fun.
Arachnids
Yes.
have gone to space.
I've gone to space
For
and
science.
Then, that I think we were talking about it, but what was so cool is that the spider made a web, and it wasn't quite right. They took it down, built another one, and it was perfect the second time.
So you're
they
saying the
learned-
spider learned.
Yep.
That's
Yeah.
That fast? It did
Pretty
it once. It was
cool.
messed up.
Yeah.
It built its web
And
again.
it knew that it was messed up, took
Yeah.
it down, and knew how to adjust and make it right the second time. That's cool, right?
That's very cool. There's a question.
Smart critters.
Yeah. So many ocean creatures have gone to space. Why so many? Is there a specific reason you'd choose marine animals?
There are different type of organisms, model organisms that there's a wide range of them, and I think just from your quiz there, you picked the marine ones, but yeah, we've.
Oh, maybe so.
yeah. We might have been a little bit
land
biased on
animals, too.
those.
California coastline.
Exactly. We love the ocean.
A couple of people are asking about the birds. Okay, there's been no eagle, but have there been other birds? And so they want to know how would they learn.
Eggs, right?
Yeah, so we've flown different eggs in space and seen how they've developed. I believe there's been some birds flown, but I'm not too familiar with the science themselves associated with that.
Yeah, and there've been flying things like there've been bumblebees and insects that fly.
Do they fly? Do they succeed in?
They do. The
flying?
bumblebee, from what I remember, there was a lot of tumbling, so they clearly knew that gravity was different. Yeah.
And they figured it out eventually. Interesting.
Very cool.
Here's another ocean creature.
Oh.
No.
The lobster.
Yeah.
No?
Tasty.
Yeah, it's
Has
not
a
even
delicious one.
Yeah. Not even for the astronauts' meals? What about this guy? What is this guy?
Is it a gecko?
Oh,
Newt?
yeah, that's
It's a newt.
newt.
Yes,
Yes.
newts
No,
from-
we've done some interesting experiments with newts because we want to see what happens with the tail as it grows in space, because there's a lot of tissue generation processes associated with it. And one of the interesting things we found out was newts grown on Earth, their tails have a downward bend to them. But the newts growing in space, because there's no gravity, the tails are perfectly symmetric and grow out horizontally relative
right.
to the animal.
And would you study something like that to understand how the human body could generate tissue?
Yeah.
Is that the connection?
Yeah. So there are these. So the way the cells actually divide are very similar between all different organisms, and so newts and with stem cells and so on. There's a lot of applicability to.
Yeah, a lot of similarities.
Yeah.
Okay. I have one more. All right, now think hard. What about this guy? All right.
Favorite.
This is the narwhal. No.
I can imagine there'd be a lot of issues with
fitting that.
Right, fitting it, perhaps. Maybe
Some certain complexities to sending a narwhal to space. We did do a Twitter poll on this subject over the last couple of days, and it seems that our followers nailed it. The narwhal has not been to space. Not yet. You guys were right, so well done. All right, that's all I got for you. So why the great diversity of organisms? Can you just kind of sum that up for us?
Yeah, so the different organisms are studied in different ways. One organism that I could talk to are the rodents that we fly in space. A lot of the mice and rats that we have in space are used for different types of experiments. A number of them are involving bone and muscle research. And so we've had experiments on space station where we want to examine what happens over a course of time with the bone that are in the mice. So what we have here is a model of a mouse femur, or the hip bone. And no, this is not the actual size of
Right.
Oh,
the mouse.
gosh.
Although I do tease kids when I show them this. I tell them that we're flying giant mice on space station, and their eyes pop open.
Massive giant
But
mice.
no, this is a 3D model. The actual bone is much smaller. It's smaller than it's just a fraction of an inch long. But the bone loss is significant in space, and the bone loss we see in mice is much faster than what we see with humans. So typically, the bone loss an animal experiences within 1 month is equal to what an astronaut would experience across a whole year. So we could see results much faster.
Yeah.
Not only are we at NASA interested in this phenomenon because we're curious about what's happening with the astronauts, but it's also drug companies who are looking to deal with, come up with treatments for osteoporosis, very significant disease affecting many people.
Right.
We're working with companies like Novartis or Amgen or Eli Lilly to develop new treatments. And one examples of what we're seeing is this is an example of what bone would normally look like.
Okay.
And you can see this is the inside of the bone.
Can you guys see that with the camera?
And normally the inside of the bone does have this cheese-like structure, but there's
cheese.
small
Swiss cheese.
pores in here and a lot of blue structure. It's just colored this way to illustrate it. But the mouse bone, I was saying, in space after 30 days, they experience significant loss. And this is what we see after 30 days. So you'll see much
in the middle.
larger holes.
Yeah, right in the middle.
Yeah. And so with less structure, more holes, that's definitely telling us that the bones are far weaker.
Okay, right.
And so we're worried about this. What's going to happen when the astronauts have to do work, perhaps when they land on Mars, and if they're going to have weaker bones, it's going to be they're much more prone to injuries and such.
Right. And you need to figure that out now, right, before they arrive. There's a question that might be related to this. What's the most notable breakthrough in the medical field that has been found through space experimentation? Is that a good example?
Yeah, exactly. We're working with companies all the time to understand how this is playing out. And then we're doing experiments in cell biology, immunology, studying how to prevent people from getting infections. So there's a wide range of different diseases that we're examining.
Cool, man. Do you want to talk more about some of the other experiments that Ames has sent to space? I know rodent research is one of them.
Right.
Do you want to
Yeah, definitely.
some of the others?
So, Sharmila, I know you are one of our resident fruit fly experts here. So do you think you can tell us a little bit about the fruit fly research that's done at NASA Ames Research Center?
Yeah, certainly. I'd love to. So here you can see this vial here is, contains these fruit flies. You can see them, you probably have seen them in the house buzzing around your rotting bananas. And they're tiny, but they're extremely useful. And so fruit flies, for example, are used on Earth to understand various disease conditions, along with, how does your immune system work? So when you get infected by a microbe, a bug, and you do feel better in a day or so, how does that happen? How does your body cope? So the fruit fly actually has an innate immune system that's very similar to humans. So we learn a lot about, immune system, the brain, how does the brain respond in a new situation? And, being in space is definitely a new situation for most organisms.
Did you say a fruit fly has a brain?
It has a brain.
That's incredible.
Like, are we
right?
talking it's got to be small.
Microscopic, yes, but
wow.
was surprisingly very structurally very similar, in the way the neurons function and stuff.
Wow.
Is actually very similar to there are many fewer neurons, there are fewer cells.
Right.
So it's simpler, which also makes it easier to study, because then you actually, can look at changes in the brain and changes in the behavior in new situations and start to draw correlations as to, which genes are important, which part of the brain is important.
Wow.
So similarly, the heart is another important, organ system. There's several of these, the circadian system, which means,
your night and day.
The night and day.
Sleeping,
So when you have jet lag and, or you have,
all been there.
stayed up all night with an all-nighter, your sleep is thrown off.
You can study that in flies?
And you can study that in flies. In
That's
fact,
crazy.
the Nobel Prize actually recently went to a Drosophila researcher who worked that all out in fruit flies. And it's very applicable. Those same genes, or many of those genes and pathways, are very applicable to humans. And that's how we know a lot about what happens in mammalian systems, along with studies in other models after that. But a lot of this work was started with the fly.
Right. Cool.
Wow.
Did you tell me that your students developed that hardware?
Yes. So I was just going to
That's so
say
cool.
that, the other major advantage of doing something like fruit flies, in addition to its similarity to the humans, is that you can fit thousands of these flies into a one box this size, which was developed, in fact, in my lab by summer students.
Oh, cool.
And then NASA, helped kind of ruggedize it and make it a little bit more robust. But otherwise, essentially, the design is that made by students. And so we can bring back thousands of flies in a box this small. And you can just, you can really study and understand a lot of very valuable systems that you couldn't do in humans or in a larger organism, like you said, the eagle, where you cannot,
Can't really fit
you can't really fit too many eagles in there. Yeah.
Wow. Speaking of, hardware developed here at Ames, I know there's also a piece of hardware up here developed by Ames researchers as well. Is that right?
Yeah, so what we have here is it's a cassette from the Bioculture system, and this is a system we developed at Ames to grow cells in space. What we have down at the bottom in pink and blue is the bioreactor, and that's where the cells actually grow. And to keep the cells alive, we have fluids that are circulating. And the fluids provide the nutrients, the gases to supply the cells and also carry away the waste products.
Wow, it looks like a really complex system.
Oh, it is, exactly. And then we have 10 cassettes, 10 copies of these that are in what's called the Bioculture system. So by having 10 of these, we're able to examine multiple samples or different samples of whatever cells we're wanting to grow.
So cool. I'm going to keep us moving because we're running short on time and we have so much more that we want to ask. And I have a bunch of questions that I'm going to save for in just a minute. This stuff is all things you've developed fairly recently and you're using now. What's next? Do you want to talk about the satellites?
Right. So actually, Sharmila, you're also the principal investigator for another very interesting experiment called the
Biosentinel.
Wow, that's such a cool name. I mean, anything with the word sentinel in it, I think is really cool.
You brought it, didn't you?
Yeah.
Did you grab the satellite
yeah.
for us?
Yeah.
Let's bring it up here.
Room here.
Oh, yes.
So, Biosentinel.
And while Sid is helping bring the model over so you can see what the satellite looks like, let me show you what the yeast cells look like. So these are yeast cells, not dissimilar at all to the yeast that you have that help make bread or beer. Those of you only above 21 would know what I mean by beer, but
Of course.
But so what you're seeing here, for example, each of these spots is actually hundreds and thousands of yeast cells. Each of these colonies was founded by one single cell. So remember how I talked to you about fruit flies and an advantage being numbers? Well, similarly, there I could fly thousands of flies in a box that big. Here I can fly millions and billions of cells in a satellite this size. And why is number important? Because you need those numbers to do statistics in science so that when you see a change, you know it's believable and reproducible. So you need a large number, right? So what are we doing with these yeast in the satellite and why is it interesting? Well, for one thing, remember, Sid mentioned that when you go deep into space, there's more radiation, right? And so we want to understand what that radiation would do to biology. What would happen to an astronaut who is going to Mars and coming back? And so we use these yeast cells who actually, or which, the yeast cells, have DNA repair mechanisms very similar to humans.
That's amazing.
So when the radiation actually damages DNA, for example, in the yeast cell, and then it's repaired, we can actually learn a lot about that repair mechanism using yeast cells.
That's so crazy how all of life uses the same things.
Exactly. From a
fundamentally, right?
tiny cell this size to the fly, to the eagle, to the whale.
Right. Yeah.
We're all, made up of very similar building blocks.
All
essentially.
these cells are going to go in that satellite, right?
It's going to go in the satellite.
And where is the satellite going to go?
Yeah. That's right. Good question. Very good question. It's actually going to go very, very far. It's going to go towards the sun. And it's going to, in 6 months, it'll be about a third of the way to the sun, which is approximately 30 million miles from the Earth's surface.
That's a long ways.
It's a long way.
That yeast is going very, very far.
Yeah, very brave, daring yeast. And we learn a lot from them about how the DNA will respond in this radiation environment, how it will, survive, and so on. And so we'll use it as a biosensor to understand the effects of deep space. And we'll have something called an LET spectrometer, which is also a dosimeter, which will also measure the radiation. So there'll be the biological effect and an actual physical measurement, which we will compare.
So why is there so much radiation, outside of, that far into space? Do you think you can kind of expand on that?
Absolutely. So we on Earth are lucky in a way, and many planets have this thing of where we have a magnetic field that protects us. It's called the Van Allen belt.
Okay.
Oh,
And
there it is
you
now.
can see
It's kind of like a donut.
That's
I'm
right.
not
You
gonna
see
lie.
a visual over there of that. And essentially this protective shell protects us from these highly energetic, highly damaging particles that would come at us either from the sun or from the cosmic radiation, and which could damage our DNA, which could damage, our cell membrane, etc., different parts of our body. But we're protected on Earth's surface. But then when you go to Mars or Moon, you're going beyond this protective sphere. And that's why, and before we send humans for very long duration missions, you want to send these sentinels as sensors or dosimeters.
Hence the name.
Hence the name
Biosentinel. Right.
Exactly.
Would you say that all of this biosciences research is kind of ultimately leading up to sending humans farther into space? Is that the final goal?
Absolutely.
Yeah,
That
most definitely.
for space biosciences, that's definitely a very
First to the
important.
moon, and then Mars, and then
Yeah.
beyond.
That's right.
Right,
Oh wow.
yeah.
That's right.
So we've got to move on to our next segment, and last with you guys, we're going to do a rapid fire. So if
Okay.
Bill can throw our animation up there, we'll get started. This is a segment where we ask you a whole bunch of questions. I'm going to fire a bunch of questions at you, and you answer as many as you can in the next minute or so, because then we have to move on to our next guests. Okay, so there are a bunch from our viewers. What is your favorite experiment that has been done in space? Question from Colin L. Well, that's Colin's question. My question was going to be, if you could do a new experiment, what would it be?
Okay, these are all excellent. I would say my favorite experiment in space was when we flew fruit flies it hadn't been flown for a number of years, and then we flew it to look at how the immune system changes and how it responds to an infection when they come back from space. And these fruit flies, the experiment was really successful, came back. We did the experiment, and we found some really cool things about how the immune system was perturbed by spaceflight, and found actually that they were perturbed in ways that were quite similar to the way they are affected in humans. And I thought that was very cool, because we learned quite a bit about the basics of what happens.
That's
to
cool.
cells.
Here's another one from Vanilla Man 47. Besides bone structure damage, how does space affect our body?
Oh, yeah, lots of different changes. Our cardiovascular changes, immune system changes.
I heard vision changes as well, if I
Yes.
remember correctly.
Yeah, that was interesting, because that arose only when the astronauts were in space for 3 months and longer.
Exactly.
Yeah.
Yeah, and so those are the gross anatomical changes, as we were just saying. But then even at the deeper level, at the cell level now, scientists are beginning to do a lot more molecular biology, and we're finding some pretty cool changes even at the subcellular level. So different organelles inside the cell and how they function, and how your genes make proteins, RNA and proteins, those are all affected.
So those are all changing.
Those are all changing in tiny ways, so that you can still survive and do your job. But at the same time, there are enough changes that you need to understand it, so you know how to handle it for long duration.
Over
in
time, what's
space.
that
You
gonna
wouldn't
do
want to
you?
send our
Yeah.
astronauts over blind.
Exactly. Exactly. Yeah.
Novistatic wants to know, do you expect experiments to be done in orbit around the moon?
Yes.
Oh, yeah, very much so.
For sure.
Yeah.
Yeah.
Well, there's NASA's envisioning something called Gateway, which could be a facility that will support experiments in space. So we've been starting to define what kind of research we'd like to do there.
Wow, very cool.
Exactly.
And got a lot to look forward to.
Yeah, for sure. Maybe one more quick one before we have to say goodbye. Do the astronauts have any involvement with any of the space station animal experiments, or are they run autonomously? From Jericho.
Oh no, astronauts actually are big. They're our partners in this. For example, the fruit fly mission that Christina and I ran recently, we had to have astronauts actually give the flies new food. They took the samples, they fixed it in preservative and froze them, so that when they came back down, we could analyze the molecular biology of the changes.
Yeah, they were like our hands
Yeah.
up there, and we owe so much to them and all their hard work
Yes.
up there on the station.
Yeah. And that's for the ISS, but then when you do a satellite mission, like we talked about by Sentinel, then it's completely
That's
autonomous.
all by itself,
'Cause it
performing
has
its
to.
Which
So
is a
we've
whole-
done
yeah.
both, but it's just two different ways of doing science, both very useful.
Yeah,
But
very
Right.
useful.
Yeah. Cool. All right. So I wish we could keep you for the whole hour, but we have more people to meet. So thank you so much for coming on.
thanks,
you.
both of you, for having us.
That was fun.
Thank you. All right. And for those of you watching, quick reminder, today we're talking about studying life in space and what kind of tools do we need to do that, and what kind of questions can we answer. If you have questions, please leave them on twitch.tv/NASA. They are clearing out all the hardware that you got to see in the first half, and we're bringing in a couple more researchers who are going to talk to us about the space hacks, genius hacks that they've developed in their labs to get research done either here on Earth or up on the space station. It doesn't always require some super specialized fancy equipment, and they're going to show us all about that coming up. All right. So, hello. Welcome, guys. So now we've got to re-meet our crew here. And actually, I thought we should start with you, Christina, because you're our host for the day, but you're also a scientist.
Right. So yes, Abby. Yes, I am the host for today, but I also work with fruit flies in the lab as a scientist.
Yeah.
And then to my right, we have...
Hi, I'm Cassie Duran. I am a postdoctoral fellow and a bioengineer researcher. I'm interested in stem cells in space and how regenerative mechanisms work.
Very cool.
Yeah.
I'm Justine Richardson. I'm a senior research engineer. So I actually built system to recycle air and water in space.
Awesome.
Wow, very cool. And,
job.
it's great to know you guys just a little bit, but we're actually going to try to get to know a little better. So we're going to start another segment called Weird Science.
Weird science.
All right, so Weird Science. I wanted to ask you guys, and also I will answer this myself.
Yes.
What is the strangest thing you've done in the name of science? And so while you guys think about it, I will start off with mine. And so my weirdest thing I've ever done with science is actually in college. I ran a catering company with 3 other friends, and we catered for weddings, for school events, for family parties, and things like that. But so we were able to cook for 300, 400 people at a time, and that was a lot of fun. But when I started working at NASA, boy, I did not think I would actually be catering for flies. So I actually make fly food every single week, up to their specs, of course. I don't discriminate against clients. And I make food for them so that they can survive and they can grow.
How weird. What is it? What's the food you're mixing?
Oh yeah, the food I make is made up of some grains. There's also yeast, lot of sugar. They love their sugar. And then we also put a coagulating agent, so it turns kind of like a Jell-O almost.
Fly Jello.
Fly Jell-O.
Christina Chung, Fly Jello caterer.
Great on my resume.
Nice. Excellent. Pretty weird.
Yeah, pretty weird.
A little weird, yeah.
Just a little bit.
So I don't have any weird science like that, but I have a weirdly dedicated to science kind of story. When I was also in graduate school, I was working on a personalized medicine project with the guy who was sponsoring my PhD, and we were trying to replicate skeletal joints so that we could create an implant that was specialized for each individual patient.
Oh, like custom fit.
Exactly. So for this particular project, we had done a 3-dimensional X-ray, or a X-ray I can't speak today. An 3D CT, or computed tomography.
Oh, okay.
Knew I'd get it eventually.
CT scan. I guess we've heard of that.
Yep.
Medical situations.
And what I needed to do to complete the implant was to 3D print these skeletal structures so I could fit the implant perfectly. And I needed a specific kind of printer and a specific kind of material. And after a lot of searching, I found a print shop that could do it. And they told me it would take about 16 hours to print. And the guy who was normally the tech for printing was not going to be there until 2 days later.
It would happen like that.
Exactly. That's how research works.
Oh boy.
So I literally said, well, I know how to use this printer. Can I stay here overnight? Can I be the one who prints this? And so I stayed for 16 hours, and I made sure the machine didn't break so that we had the printed model ready and we could fit the implants perfectly.
So if I'm able to ask, what did you print? What skeletal model did you print?
So for this specific project, we were looking at the jaw, or this specific section here. It's called the temporomandibular joint. When it goes bad, it goes really bad really quickly. And we were creating a tissue-engineered implant to basically correct that problem.
Wow. And then was the patient all right?
So I wasn't the doctor, but from everything I know, it went really, really well.
Oh,
Yes.
well, that's... Wow.
Your dedication paid off. Your sleepover
weird
at the
dedication.
coffee shop paid off. Excellent. Weird dedication, weird catering. Mm-hmm. What about you, Justine? Can you top that?
I don't know. Well, I can describe some of the interesting experience I have.
Yes, do.
So when I first got to Ames, one of my first job was to develop a wastewater recycling system. That goes from wastewater
Nice way of saying
actually
something
to
else.
potable water.
see where
And
this
so
is
when
going.
it was time to test the system,
Yes, how do
I
you
needed
test?
a 55-gallon barrel of urine and dirty shower water.
Boy, oh boy.
That's a lot of gallons. That's more than I make for my flies. I'll tell you that.
That.
Well, you know, we have a 5-gallon glass bottle in the men's bathroom.
So very realistic
with
is
a
what you're telling
funnel
me.
on top, and they contribute. And for the female, yes,
Such a great.
for the female, you know what? I had to go around with a bottle, an example, and say, "Can I have your urine, please?"
Great conversation starter.
It's a huge contribution to science,
Oh, it
right?
is. Oh,
You're
yes.
gonna be in the history book forever. It's a NASA document, and so that's how we got it. And we did that for weeks to try to mix this, mixture of urine. I had a fully gown.
Wow.
Yeah.
And then you used it on your system?
Yes, we did. Yes,
How'd it
we
go?
did. Actually, we did go all the way to potable water.
Wow.
Yeah, so
You
that
could
means
drink
you can
that
drink
stuff,
it.
right?
We
Okay.
didn't drink it.
Oh, okay.
But you could have drank it.
I could have drank it. Yes.
There's an important distinction between the two. I'm just going to say that right now.
Yeah.
That's pretty impressive, though.
Yeah, that was not my preferred job.
But I always say, like, for the science.
Yeah.
Am I right?
Yeah, in the name of science.
You got to do some
Yes.
weird things to get the NASA job done, I guess.
That's right. Yeah.
So, yeah, that is completely weird, Justine. But,
Very interesting. Yes.
but I guess what's not weird is using everyday items to get your research done, because that's what you guys are going to show us in the next section, right? So we're going to move on now to present to you some of our genius space hacks. So as I was describing before, these guys sometimes encounter challenges in the lab, and they need quick fixes or they need smart fixes, and you don't necessarily need some super fancy equipment to do it. You can invent something with a little creativity. So, Christina, let's start with you and your flies. This first hack we're calling Lord of the Fly Sorters. You are a fly sorter extraordinaire. How many do you think you've sorted over the years?
Well, Abby, that is extremely flattering
well.
title. One of the fly sorters. Yes, I have sorted many flies in my lifetime. I would say tens of thousands at least.
Good Lord.
Yes, it's a lot to think about. It's a lot of hours, but yes.
Why are you sorting flies? Tell us where this all begins.
Well, that's a great question. So why do I sort flies? Actually, believe it or not, flies are actually they're different. They're not all the same. They might all look the same, to the naked eye, but they're actually a little bit different. And we do this so that we want to make sure that we know for our experiments, what fly goes where, essentially.
That makes sense.
And so one of the most common
I can see them actually.
you
I just
see them?
realized in the close-up I see them.
Yeah, they're all happy
scurrying
up there,
around.
scurrying around at top.
Okay.
So actually, one common way that we sort is actually between males and females, and so they are distinctively different, and we're able to see that under a microscope. And so normally, what we do in the lab is that we actually take flies that are in a vial like this, and we'll have a tray kind of out. Just about this big, about a cell phone sized tray or so. And then we'll actually dump out the flies and they'll form a little pile. And unfortunately, we can't just tell the flies, like, guys on this side, girls on this side,
Right.
and just have them all lined up. Unfortunately, we can't even get humans to do that. So
Yeah.
it's not going to happen for flies. So, how do we do that? Well, actually, what we do is we use...
There it is.
Oh, yep, and then she's actually there
A
using the
paintbrush.
hack. I don't know if you can actually see it on there, but she's actually using the hack.
A
and you can see...
paintbrush.
Yep, a paintbrush. And you can see on the left is actually females, I believe, and on the right is males.
Wow, you can tell just by
Yeah,
looking
I
at
can tell. It's a seasoned eye. They can tell. But you learn after about a couple thousand flies. You'll start learning.
Is that all it takes?
That's all it takes. A couple thousand flies here or there.
Okay, so you just need an ordinary watercolor type paintbrush
Yes.
to sort flies for space station experiments, basically.
Yes, exactly. And this is my favorite paintbrush. It is not only because it's a beautiful color, it's pink and magenta color, but also it has actually very sturdy bristles, but actually still very soft. So what we can do is actually pick up individual flies.
Oh, wow.
And place them where we want them to go. And we're also able to do like a sweeping motion to kind of move the flies into different piles.
Wow.
Genius.
Amazing.
You don't
Yep.
need some specialized fly sorting device.
Nope, we just need to go to a
paintbrush.
craft store or a watercolor set if you take it from a watercolor palette, and you can use that for this.
Awesome. So Digital D has a question for you.
Oh, yes.
Have the flies ever escaped?
Oh, yes. If I'm going to be honest, then yes, they have escaped. But mostly that's a user error more so than the flies. But what we do is we're able to either kind of catch them and scoop them back up, or sometimes we just let them go.
Farewell.
Farewell, flies. I'm sorry you can't join us in our experiment.
Right.
So yeah.
All right. So those are the experiments that Sharmila was talking about a little while ago.
Yes. So
studying.
I actually work in her lab. So she is someone I'm very close to, and we sort flies all the time.
Fascinating. Now, as weird as that is, I just want to mention that someone has voted in Justine as the winner of Weird Science.
I agree completely with that.
I don't mind that.
Whoever it is,
She
would
can
you like
take
to
that.
volunteer?
She can definitely take that.
I'm hiring.
Now, as we move on to our second hack, a little shout out to I Am Lungund, who mentions, I put fruit flies to sleep with sleeping gas in biology class. Stay tuned. Here comes hack number 2, which we are calling Nighty Night Flies. Now, that's not a nursery rhyme that you missed in your youth. That is about a way to make flies fall asleep fast, right, Christina?
Yes, that's definitely very true.
So tell us, how does that normally work in the lab, and then what's your hack all about?
Yeah, so normally in the lab, actually what we do is we have a big carbon dioxide tank. If we want, we can cue up the photo.
Yeah, there's a photo of you next to it, I think. So why carbon dioxide? Because that's going to knock out your flies.
Oh, there we go. So carbon dioxide will actually put the flies to sleep. And when I say that, I literally mean put the flies to sleep, not put the flies to sleep.
So they wake up.
Yeah,
They can wake up,
wake up.
And most of the time we want them to wake back up. That's
Right.
actually the goal of this. It's supposed to be a
temporary.
temporary thing. And so what we do is, so we actually have to do this normally. This is how we're able to maneuver the flies with carbon dioxide when they fall asleep. And so, yeah, that's why we have to use carbon dioxide.
All right, but you were telling me then, okay, so the fly experiments go to the space station. They spend perhaps a month there, right?
Yep.
And then some of the flies come back for you to study changes in them
Right.
back on the ground, right?
Yes.
And then is that when you're going to need a hack? Because you have to go fetch them.
Yes. So actually what we do is we actually have to go to Long Beach, California, which is down the coast of California. Since we're up in north, it's actually down further south. And so we can't just, strap a tank to our car and expect to drive down with it.
You don't want to go down the highway with an 80-pound tank on the roof?
No, and with those potholes in California, for all of our Californians, we all know this. Yeah, that's just not going to fly.
Fly.
Fly count pun number 1. So no, we're not going to be able to do that. And so what the genius scientists at NASA Ames Research Center did was actually found a way to create portable carbon dioxide.
Okay.
Yep.
How do you do that?
Yeah, no, that's a great question. So actually what we did was we have a bottle. And this is a fly bottle that we have in the lab all the time. We hold flies in it. It's kind of like this vial, just bigger, and so we can hold more flies. And so what we did was we took a spare one, and we filled it up with tap water, just water you can find in the sink. And then what we did was we actually were able to hack this by
What is that?
dropping 2 antacid tablets. And so,
Antacids.
yeah, antacid tablets. So this is actually, antacid tablets are used for indigestion. Those are the ones you can find at your local drugstore.
Really?
Yep, these are just the same ones. And so these 2 tablets. And so to demonstrate this hack, we're actually going to be able to show you how the flies fall asleep. But what I also wanted to make sure was that safety first. Yes, so
yes.
we all
have glasses.
have
There we go.
goggles.
All right, goggle up.
Goggle up.
Now we're geeky.
Science to do
There's
here.
nothing wrong with looking geeky. NASA is very concerned about everybody's safety. Always, including on set. And so, what we did was that we have a bottle of water, And then now we have a vial of flies. And so I'm going to drop these 2 tablets, and then can I get a quick countdown from the folks here?
3, 2, 1.
There we go.
And you can see it's
yeah, you see the
actually
bubbles.
fizzing, right?
Yeah, I see a lot of bubbles.
And so what we do is now we have to stop it with a rubber stopper, and now we're actually able to direct the carbon dioxide out the needle. And what we can do is take the vial. You can see they're all crawling up there, getting all excited. And what we'll do is we'll kind of insert it in, and we'll wait for the carbon dioxide to affect them. And as you can see,
Yeah, they're starting to drop.
they're starting to drop.
That's pretty quick.
They're dropping like flies.
Oh, here.
Oh,
punch
sorry.
number
Pun number 2.
Wow.
I'm
Like
sorry, I can't help
flies.
myself. So as you can see now, they're all asleep at the bottom. You can see them right there. And so yeah, now we have about 30 seconds or so to be able to sort the flies, much like we did before at the hack. We bring our trusty little paintbrush and separate the flies into their appropriate containers.
That's not a lot of time, though.
No, we have some trained hands in the team that we can
Yeah.
work really fast and very carefully, but accurately as well.
Interesting. And while we're talking about working with flies and other organisms and putting them to sleep, letting them wake up, what can you guys tell us about the precautions you take to treat animals correctly and humanely?
Right.
There are rules around that, right, in your work that...
Oh yeah, definitely. And I know, I speak for myself in my own research, but I know these folks, especially Cassie here, also is very familiar with this as well. But yes, we take the utmost precaution and utmost just sensitivity to the animals, and knowing that they are giving their lives for us to be able to learn more in science.
And they live good lives.
Yes,
they get to lay around, they eat a lot, they chill out all day in a nice warm room. They're
Right,
perfectly happy.
Catered food.
Yes, catered food. Yes, I know all about that. And so we take here at NASA, especially, we take very much care into that and making sure that we don't do things just for fun. We do it for
right.
the advancement of science and
Yeah.
to help help us as humans as well.
Right. Right.
Yeah.
Thank you for that.
Of course.
Really neat.
Right. Fascinating. So I hope that the viewer who has done that before got a kick out of seeing a NASA scientist putting flies to sleep, probably in a similar way. Let's move on, though, to our next hack, which Cassie is going to walk us through. So this is about sending spacecraft into space, such as satellites, and before NASA launches anything and sends it up there for good to run an experiment, they're going to test it very carefully and give it a sort of spacecraft checkup, which is the name of this next hack. Cassie, can you tell us what's the deal and
Sure.
how do you
I can go into this a little. So when we package our experiments for flight, they're usually contained in these nice sturdy boxes so that if anybody drops it, or if it needs to get on the space station, there's no gravity, so we orient them upside down. Sometimes they're attached to the walls, sometimes they're in the floors, they kind of go all over.
Okay.
So we pack them up in these nice sturdy containers, but once they're packed in here, we can't really check on them to make sure that everything inside is still working. So how do we do a go, no go, basically, for flight on an experiment?
So, how do you
this?
decide this is
Yeah.
good to go, it's working correctly,
Yeah,
or
because
stop,
of
it's
course
not?
at NASA everything has a backup and then a backup to the backup.
Okay.
So we want
Right.
to make sure that we're sending up a working model.
Yeah.
So before we actually put things together, we do a last minute checkup, and checkup is an accurate word because we use a trusty stethoscope.
I've seen those before.
Yep, there you go. Anybody who's ever been to a doctor has seen one of these before. So literally, if I plug these into my ears, I can take my stethoscope and I can listen for moving parts within, this system. Sometimes it's a pump, sometimes it's check valves, sometimes it's gas going through the system.
Oh, wow, you can hear
and
all
I
that.
can hear all of that by moving around my stethoscope to make sure everything's in working order.
Awesome.
So what are you listening? What kind of sound are we listening for? Can you?
Yeah.
describe a little
So
bit?
a valve will be a click click.
Oh, okay. Oh,
Yeah.
so okay.
Literally, as the valve opens, you'll hear it open. As it closes, if there's any gases running, for example, you'll hear a hissing noise, just like we kind of heard a little bit when your CO2
Right, with
was
the little
going into the
with
flies.
the carbon dioxide. Yep.
Exactly. So I'm going to
That's so cool. Why reinvent the wheel or
exactly.
the stethoscope
It's already going.
when that thing already exists to do what you need
So
to
Cassie,
do?
can we call you a space doctor?
Please don't. But yeah, that would be too cool. I'm not quite there yet. Not cool.
All right, let's move on
right.
to our next hack, which you're also going to show us, so we'll give you a moment to get it up here. But I think you're going to tell us about spinning for science and space. So we're going to call this hack Spin Class. So this is about using a centrifuge, right? So I worked in the lab briefly. I remember what that is. Can you run us through, though, what a centrifuge is, how that works?
Sure, absolutely. So one of the major things we have to do in research is we have to be able to separate our samples. Just like Christina uses her paintbrush to separate flies, we have to separate cells from culture media, from other like water components and things like that.
Yeah, okay.
And the way we do that is on Earth, you know, for example, if you wanted to pour a glass of water, gravity kind of does the work for you. You pour the glass of water and you're fine. In space, however, because there's no gravity, we have to use other forces to drive fluids in certain directions. And one of the main ones we use is a centrifuge. Centrifuge is basically a rotary device that spins and creates a load or a force in a single direction. And with that single direction force, you're able to separate liquids based on densities or masses, or move fluids from one position to another.
Okay.
Oh.
So on that note, I think I know what that's all about. I happen to have brought my salad spinner with me
boy,
today.
I am
so
hungry.
Is this what we're talking about? I've got my wet lettuce. Throw it in here, right? And then I'm going to get this spinning. Right?
More or less, that is exactly what we're talking about.
And it's going to kind of throw the water against the wall, and that's going to drain out the bottom, and my lettuce is dry. Is
Perfect.
that
Exactly.
the same idea?
It's
as
the
your
same
centrifuge?
idea. The difference is, if I'm separating out microscopic cells, I need something a little bit more powerful than your arm.
Oh,
Sorry. You look
Fine.
strong, but
fine. Whatever.
Although we could use salad.
we could use salad. Yeah, is there anything in there?
So I'm happy to chow down.
So all right. So something a little more powerful than this.
Exactly.
But a lab centrifuge is a big thing, right? You brought a picture.
We actually do have a
Yes.
photo, so you can pull that up.
Oh, wow.
All right. So it's a big machine. That's
Yes.
not a thing that you can easily launch to the space station.
No, definitely not. This thing would take up way too much mass and way too much space. It's a big machine. So basically, we needed a version of that we could then use in space that wouldn't take up really any excess space, and that's our second hack,
All
or
right.
our third hack, I
Ooh,
guess.
Bring it out.
So,
Bring it out.
how do we do that? We use dun dun dun.
Oh, my
Power
goodness.
drill. A power drill. This is a very traditional, like the astronauts will use. They have a couple of versions of this on station, but they'll use it for repairs. They'll use it for repositioning of gear on station. And we decided that we would use it as our makeshift centrifuge.
All right. Cool.
And the way we do that is we have a drill, and we used a 3D printer, and we printed one of these. This is basically a centrifuge rotor. And what this does, get a zoom in on that. What this does is it has several positions along the rim where we can put sample tubes. And I have an example of a sample tube that's out right now. So this is a sample tube.
That looks kind of interesting.
And our sample tube fits right into this rotor. So if I pop it open a little bit, bring it closer so I can see what I'm doing. So if I open the lock, I can pop the tube right in. If I lock the tube back in, I can then mount this whole thing. Let's see if I can do that. Yep, I can successfully. And then this, if I had my sample in, I can literally run this. And I would like to run it, but safety first.
Oh, my gosh.
Bum,
No, we're
bum.
going to need another.
You
Okay.
guys are better than me. I forget every time.
That's because we work in a lab.
we can't forget. They don't let us
So
forget.
if I have my centrifuge, this will spin nicely. I'm going to spin it slowly because we're in a studio.
All right.
But yeah, and basically after this is finished spinning, I have a photo of what happens with some dyed liquid. And what you can see here, if we can see, there we go. We have two pictures. This picture over here is before the sample is centrifuged. So you can see that all the sample is in the loading input well. And then after centrifuge, the sample has moved down and is now in the test window.
Cool.
So now from this state, we could do whatever kinds of tests we're actually
right.
looking to do.
Let's see that video of astronaut Kate Rubins
yeah,
using this
absolutely.
hack on the space station.
All right, so she's.
There she is. That's exactly the same thing you just showed us.
Exactly the same thing. Same drill even.
All right.
And she's having just as much trouble. And there it goes.
She's spinning.
And she's spinning it at full speed.
Cool.
Wow, that's pretty neat.
Awesome. I love that that's actually in use up there.
Yep,
That is so
in use.
cool.
And she's
she's
obviously
very
loving
happy.
it.
She's
Yeah.
it's working.
Okay.
That they're happy.
Well, we've got to keep moving because we honestly have like 4 minutes left, so we have to say goodbye. So we want to hear about Justine's hacks. So this next one we're going to call take my astronaut breath away, because Justine works on keeping air clean for astronauts on the space station. And I know that you've helped design some systems that are already in use up there, and that you have some new research going on that you can tell us about. Can you tell us the basics? What's the deal? We have to clean up the air that they've exhaled, the astronauts?
Right. So we take all the information that you guys are doing and learn from it. And on the space station, as the astronaut breathe in oxygen, they actually breathe out carbon dioxide. We need to reduce that carbon dioxide in a room like this to a level that's reasonable. So one way we do that is we use a material called solid sorbents.
Sorbents?
Yeah,
like absorbents?
Absorbents.
Yeah.
Now, if you take a look at these sponge, you put this in water is absorbed, and you squeeze it out using pressure, or if you put it in the sun, the water comes back out. Another example of solid sorbent is like that silica gel bag. Where do you find those?
Oh, I've seen these in new shoe boxes, definitely.
Right.
Beef jerky?
Yes.
Yep.
Beef jerky?
Beef jerky.
There we go.
You can eat your beef jerky, but do not eat the silica
The
gel.
silica gel bag, right. So we use a material that removes carbon dioxide. Just like your water filter at home, remove contaminant from your water. So in order to remove the carbon dioxide, we have to first remove the water. So we use a similar experiment material, which is silica gel on top.
So the same stuff in that packet is in this.
Not same, but similar.
Okay, in this
right.
space station
And
air
then-
recycling.
Right, you flow air through. The space station, the system is bigger, it's like 2 5-gallon barrel, and then the air stream is dry. When it's dry, then we remove the carbon dioxide.
And the carbon dioxide is removed by these dry parts.
Yes, by the second sorbent that
Very
remove
cool.
carbon
And is this reusable too?
That is reusable because, you put this in the sun, the water evaporate, you heat that up, the CO2 then comes off and it gets recycled and converted to methane and water.
So we waste nothing on station.
No,
you
everything is reusable, regenerable, but it has to be safe material, right? Mm-hmm. Can't use a lot of chemicals.
All right.
So what's some of the current research that's being done on
So
this
some
sort
of the
thing?
more advanced research, as we travel a little bit further from ISS or International Space Station, we look at some other sorbents that are more effective that they use in the submarines sometimes.
Ah, cool.
And
Submarines
so
and
these are
spaceships.
these are called liquid sorbents.
Liquid sorbent. That's cool.
So an example of this is the material on top right there is a liquid sorbent that absorbs CO2 when it's mixed in water. And when you add CO2 to it, it actually becomes one phase.
That's cool. So this looks like oil and water, or like salad dressing.
Right,
salad.
for your salad.
Salad,
Yeah.
yes.
Yes.
Then it absorbs CO2 and it becomes mixable.
One mixable. And
Yeah.
then you heat it up and it will reverse right back.
So cool.
So it's regenerative as well.
It's regenerative,
and
reused.
reusable.
That is
Cool.
the key word. Right.
Incredible. I'm
Definitely.
impressed. You guys, so clever. And I am so sad to say that we have to say goodbye. These conversations go so fast every time, but thank you, all of you, for joining us. And thank you, everybody, for watching. This has been NASA in Silicon Valley Live. As we said, we are a conversational show about all the cool work going on at NASA's Ames Research Center. So tune in next time. We will be back on September 27th to talk about video games in space. So join us then. You can watch us on Twitch, YouTube, Facebook, and NASA TV, and you can catch the audio version on our podcast. Until then, thank you so much for watching, and thank you guys for joining us.
Of course.
Bye.